One of the strengths of top-down proteomics that really drives the field is the ability to look at what we call proteoforms. And proteoforms are nothing more than differently modified proteins that come from the same gene. So a gene can be spliced and create proteins of different sizes and so forth, and then they can be differentially modified depending on what function they might have. And so one of the key elements of top-down proteomics is to try to define how is the function changed by various modifications to a protein or patterns of modifications to a protein.
Hello and welcome to the PitCon podcast. In this episode, we'll be speaking to John Yates about the role of top-down proteomics in capturing cell-to-cell heterogeneity. Thank you for joining us here today. The first question is, please could you introduce yourself and briefly describe the main focus of your research?
My name is John Yates, and I'm at the Scripps Research Institute, which is about 20 minutes up the road from here. And the focus of my research has been on protein analysis by mass spectrometry for 40 years.
And what inspired you to first focus on this area?
So I was a zoology major in college, and I really loved biology, and I got interested in sequencing proteins because there was a group where I was in college that was using mass spectrometry to sequence proteins, and I thought that was just the coolest thing ever because there was actually a computer on the mass spectrometer, and computers were new and they were exciting, and so that's what really kind of drove me into this process of trying to sequence proteins by mass spectrometry.
Perfect.
It's a lot harder back then.
And your research emphasizes the importance of understanding cell-to-cell heterogeneity. For those who don't know, what is meant by that term, cell-to-cell heterogeneity?
So cells are different, particularly if you look at a tissue. Tissues are, you can think about a cell as a system. And so tissues are just systems of systems, a system of systems. And so you want to try to understand the differences between cells and even the differences between the same type of cells. Because they're reacting to their local environment and things that are going on locally. And so we want to try to understand all of that.
Yeah. And how did you initially identify this particular area of study as one that you wanted to explore in terms of the context of health and disease?
So it was a natural evolution. So as the techniques for doing proteomics get more and more powerful, it just made sense to try to study different aspects of biology. So we rapidly moved from cells to tissues to whole organisms.
Perfect, thank you. And can you elaborate on the significance of cell-to-cell heterogeneity in understanding biological functions and disease mechanisms?
One way to think about it is that you can have mutations in proteins, and in one cell type, that mutation is devastating. And in another cell type, that mutation is just a passenger. So it doesn't affect the biology of the cell, and the cell happily moves along, doing its business and so forth. A good example of that is ALS, Lou Gehrig's disease, where the mutations that occur in the protein superoxide dismutase are so damaging to motor neurons that they die and people start losing muscular function.
And what are some of the challenges and potential advancements in applying top-down proteomics for individual cell analysis?
So the field of proteomics mostly focuses on digesting proteins to peptides. And part of the rationale for that is that the technologies to analyze peptides is more mature and more robust. The technology to analyze proteins is not as robust, and proteins are actually much harder because the chemistry of proteins is more diverse, more extreme, one could say. There's a big difference between membrane proteins and which are very hydrophobic and very difficult to deal with, and proteins like mucins, which are very, very heavily glycosylated. And so dealing with intact proteins, there are many more challenges that you have to deal with. And so that makes top-down proteomics has lagged bottom-up proteomics in its ability to really address important biological questions.
Yeah. What is, for those who don't know, what is exactly meant by top-down proteomics?
It's very simple. It just means that we're analyzing an intact protein rather than digesting it up into peptides.
Yeah. And how does this technique in particular help to better define heterogeneity between cells?
So one of the strengths of top-down proteomics that really drives the field is the ability to look at what we call proteoforms. And proteoforms are nothing more than differently modified proteins that come from the same gene. So a gene can be spliced and create proteins of different sizes and so forth. And then they can be differentially modified depending on what function they might have. And so one of the key elements of top-down proteomics is to try to define what is the function, how is the function changed by various modifications to a protein or patterns of modifications to a protein.
Great.
And that's best done by top-down proteomics.
Great, thank you. And in your approach using shotgun top-down proteomics, what are the key advantages of employing a cell-in-one device and ETHCD over traditional methods?
So the cell-in-one is a new device that is very good at isolating cells. And I don't actually have one in my lab. I collaborate with the group that has one, and they've figured out how to analyze how to isolate cardiomyocytes. And it's got this nice video feed on it so that you can see the cardiomyocyte. And they're actually still beating as they pass through the system, so you can tell that they're still alive. But what the cell-in-one has done, which has really been quite revolutionary, is it allows us a new way of isolating cells that's different from the standard method, which is flow cytometry.
And you've mentioned there the cardiomyocyte cells in particular. What were some of the biggest challenges of using that particular method for analysing these cells?
Yeah, we haven't solved all the problems yet, but cardiomyocytes, the contractile machinery in the cardiomyocytes, so the proteins that are making the cardiomyocytes beat, are not very soluble. And so you have to work out methods to try to get them soluble enough that you can get them into the mass spectrometer so that you can measure them.
Thank you. And your study identified a high degree of proteoform heterogeneity among individual cardiomyocyte cells. How does this finding alter our understanding of cardiac biology?
Well, it doesn't yet, but it's on the pathway to allowing us to try to. One of the things we want to understand is there are cardiomyocytes of different size, ranging from 40 microns to 100 microns, and we want to understand, are there biological differences between the sizes of the cardiomyocytes? And those sizes may, in fact, change as a function of where they're located in the ventricle of the heart.
That's really interesting, thank you. And you mentioned in your talk the mention of discovery of numerous post-translational modifications such as phosphorylation. Could you tell us more about this finding and what implications these findings have for the future of cardiac research or therapies?
It's still probably a little bit too soon to get to understand if those findings have any relevance to the biology. But it's what's important is that we now have the ability to measure those things, and so now we can look at not only normal hearts but start looking at diseased hearts and see how the phosphorylation patterns might change as a function of disease. And there have been some studies which have been done on not single cells in cardiomyocytes but on bulk cells. And they have seen that there are indeed changes in phosphorylation patterns as a function of heart failure. And so trying to understand what drives that can help us think about new therapies.
Yeah. And so leading on from that, what are the next steps for your research?
Well, we're going to try to improve the analysis in a couple of ways. So we want to be able to get access to improve the analysis so we can look at bigger and bigger proteins within the cardiomyocytes. But we also want to be able to fragment the proteins better so that we can get more reliable information on where modifications are located in each of the proteins. And we want to be able to analyze all the proteins from the cardiomyocytes. Right now, we're only getting a fraction of them.
PitCon Perspectives. Unforgettable PitCon moments from the experts.
So, as we mark the 75th anniversary of PitCon, could you share your first memory or experience of attending this conference and how it impacted your view of the scientific community?
I think this goes back maybe 30 years. I was walking down to the exposition hall was mind-blowing, because there's just everything out there. Anything you would need to do science was on the exposition floor. And the conference was huge back then. There were 30,000 people, and there's a lot of excitement, and a lot of the vendors were having special events all around. And so it was super exciting. I was there. I think my first PitCon was because I was on the editorial advisory board for the journal Analytical Chemistry, and they held their meeting at PitCon because so many people would go to PitCon for the science. And so we held a meeting there. That was my first exposure to PitCon.
And finally, what are you most looking forward to about PitCon 2024 in San Diego? Or, because we're sort of a few days in, what have you enjoyed most about the PitCon 2024 so far?
I didn't have to travel to get here, so that was nice, and it was what was fun for me was being able to show my friends my city, and so be able to pal around with my colleagues and friends throughout the area of downtown San Diego.